Hello to all of you who are interested in Python program. I already started asking for your like and your subscription to this channel. This is the fourth video and the subject will be genetics. The definition of a genetic code is the relationship in the protein between the sequence of bases in DNA and the corresponding sequence of amino acids. In other words, the genetic code of DNA is expressed by broken bases which were called codons, which codon formed by 3 letters correspond to a certain amino acid. Let's recall a structure of DNA. As nitrogenous bases that form the DNA are adenine, guanine, thymine, and cytosine. A nucleotide is composed of a phosphoric acid, a sugar, and a nitrogen base. A sequence of DNA nucleotides form a gene. GenBank is a public database of nucleotides storing information on nucleotide sequence for thousands of species. The GenBank file format includes information on nucleotide sequence and their translation into amino acids. Through GenBank, we will select some nucleotide sequence and translate them. We will make the program in Python, compare whether this translation is the same as the one provided on GenBank. Let's start by accessing GenBank on the site www.ncbi.nlm.nih.gov. Click in All Results. Now access GenBank. At the top, write the name of the nucleotide XM_011242420.3. This will be the nucleotide that will be worked on. The gene has this size 1 to 2744. In CDS, the size of the translated gene is entered 321 to 1580. See the translation at the bottom. One very important thing that you have to keep in mind is the DNA stop codons. They are TAA and TGA. If they show up, we will put on underline. See TGA as the last codon. You will then need to put an underline at the end of each translation. Will we be able to achieve this same result? This is what we are going to check. The first step is to save the translation in the script and call it a codon. Afterwards, save all this orange file in txt without the numbering. Write the program address in input file, ask the program to read the file and merge the lines. This nucleotide source file will be called a SEQ. You need to select the string that was translated from 321 to 1580. Remember that Python does the numbering from 0, then the selection would be from 320 to 1579. To select up the nitrogen base 1579, you must enter 1580. Then ask the program to read the data in sequence from 320 to 1580. Let's create a function called translate that will translate the SEQ database. This part is the translation of each crack to the corresponding amino acid. I will leave some bibliographic reference in the information box to search for this information. What we need now is to select each crack in the SEQ database and classify it according to the find table. New SEQ will be the translation of the SEQ database. Let's compare if new SEQ and column, which is on the website, are the same. When the program runs, we'll see that it does. Equality went true. How about repeating this with another nucleotide? Type NM_1386-180.6 in nucleotide. Going in CDS, we find that the size of 150 to 1214 has been translated. Then, Python should read the source file from 149 to 1214. Write the nucleotide translated by GenBank in codon. Note that the final codon is TAA. Put it when copying the translation. Save the source file in txt without the numbering. Now SEQ will be a part of the source file from 149 to 1214. If the programs run, the program translated the code perfectly, as it is in GenBank. Very interesting to be able to use Python in the field of generic, isn't it? Did you like the video? So, I wait for you next time. See you later. Bye!